I was not expecting that. Good evening, everyone. Good evening. How are you all? Good evening, everyone. I hope that you can see me and can hear me clearly. Good evening, everyone. Welcome to the class. It's your own teacher, Ankita. Good evening, everyone. Hi, I can see all of you here. How are you all? Great. Good to see you that you are here. And we are really sorry there was some issue, but now it's all fine. And thank you for waiting for us. Super happy. Fine, I can see a lot of you are here. I'm super excited for an amazing class today. And today is the all cell organelles that we have in the cell. So till now, our journey has been really very interesting, right? Yes. We had a very amazing journey. We started with the cell, right? Then we talked about the important components of it. We talked about cell membrane, cell wall, nucleus, and finally, we are here with the different cell organelles. Mayank, today's session is for you. Yes, I can see that all of you are excited. Let's quickly start our session. I want all of you to pay attention, have your notebook and a pen or a pencil, right, so that you can note down the important points that we'll be discussing. Awesome, everyone. Very good, yes. Mona and Lisa, we will be doing that, but not in today's class. Awesome. I can see that all of you are here. Quickly give me a thumbs up and we'll start. Right? I hope that... Yes, very good. I hope that all of you have joined our Telegram group, right, where definitely we can communicate. You will be getting the revision question and most importantly, the session notes, right? The session notes, so that you have a quick revision of the things, right? This is really very important. Very good, right? And of course, you'll get the session updates. You have homework questions over there and interesting quiz will be happening over there. Awesome, everyone. Very good. I can see many of you are saying you've already joined. Very good. The link is in the description box below. You can go and check that out. Awesome. Now, let's quickly see the content we have. Now, today is a lengthy class. I will not study for 1 hour, but you know what? We will be learning for approximately, I would say, 45 minutes, right? So, I want you to have a clear check. For 45 minutes, we will be focused and we'll be learning. Yes. Right, everyone? I'm in the studio, yes. Great. Awesome. So we are clear with that, right? And over here, as you can see, this is the order we will be going ahead. So I want you to have some patience. When I'm teaching you mitochondria, please don't ask me to teach about the lysosomes. Okay? So you know the order. We have mitochondria first, then we will be learning about ER, Golgi bodies, lysosomes, vacuole, and the plastids. Right? So we have this, right? And let's quickly start. Before we start, let's quickly clap for our homework rock stars, right? We have to appreciate them because they're doing amazing. Let me quickly call out the names of our friends who wrote the answer, right? For yesterday's question. It was a very easy question though. What is the difference between the chromosome and the chromatin? Over here we have so many of our friends have written down the answer. So we have Zarina, Himani, Prachi, Prem Kumar, Archit, Sharanya, Chitra, Kashish, Sunena, Nishu, Ritu. Claps for our friends. Just 1 minute, right? Let's appreciate our friends. Ritu, we have Divyashmi, then we have Swati, we have Venkat, we have Aditi, Blackpink, we have Rashmi, Shrika, Gayatri, Zarina again have mentioned few more things, very good. Prashansa, Tanisha, Vartika, Om Shankar, Sai. We have Abhinay, we have Monalisha, Gauri, Kanika. Oh my God, I'm super happy to see all of these answers, right? And you know what, when I was reading the answers, it feels that, it's very good that you know what you are writing. With this, you're actually learning the skills, right? That you know what are the important keywords that you have to mention in the examination, right? Ayushi, you can write the answers in the previous video, right? So for example, this is a video. Once the video will be over, right, when the class will be getting over, after that, in the comment section below, you can mention your answer. It's okay, Gargi, no issues tomorrow. If you haven't attended that question, that's perfectly fine. Anushka, I'm sorry, yes, I missed your name over there. No issues. Very good, Abhinay. Abhinay is mentioning the length of the DNA in one cell. Very good. So I see that you did a lot of research. So are we excited, everyone? Today also I will be giving you some interesting question. Divyashmi, I've answered you. I called out your name. Yes, very good. So quickly give me a thumbs up. Very quickly, right? A quick thumbs up, everyone, or a smiley or anything. Let's just see how excited are we, right? And ready, get set and go. So till now we have studied about the cells, right? We have studied about the two different cells. We have plant cells and we have the animal cells. We have discussed about the prokaryotic cells and the eukaryotic cells also. And we are clear that these two over here, right? Plant cells and the animal cells are what? Whether they are eukaryotes or prokaryotes. Quickly tell me. It's a very easy answer though. Very quickly. Yes, Jeevani, yes, I can see the shock on your face. Yes, Blackpink, we have talked about the cytoplasm. Very good. I can see over there, we are the eukaryotes, right? These two are the eukaryotic type of cells because we have the nucleus intact. So as of now, till now, we have discussed about the nucleus, cell membrane and cell wall, right? And of course, the nucleus we have studied yesterday only. And now we'll be discussing about the mitochondria, ER and other things. So let's start with it. We are starting with the mitochondria. Now, no doubt we know what mitochondria is. It's a very, very important cell organelle, right? In our cells, we have mitochondria, which of course has double membrane. That's a very, very important point that we should remember. Mitochondria have double membrane. So if you look at the structure of the mitochondria, there is an internal membrane and the outer membrane. Inner membrane and the outer membrane. Then of course, very interesting thing about mitochondria is that they have their own DNA. Can you imagine these little organelles that are present inside the cells? They have their own DNA and own ribosomes. Means that they are capable of making their own protein, first of all, and they can actually self-replicate also. Interesting thing, right? Yes, I can see a lot of you are saying east or west, Byju's is the best. Thank you so much. It's all your love. Thank you, Hari Arjun. Anuj, yeah. Yes, we'll be discussing about that. Very good, very good. Sandhya, I would request you not to spam. I will be taking up that answer, but not now. Yes. Very good, Om Shankar. We know that they have the DNA because the mitochondria were initially long back. They were all alone. They were capable of living outside, right? And they were like kind of one man, one organelle that is doing all of its function. Slowly with the due course of time, of course, we are here. Right, everyone? Yes, Ritu, just give me a minute. We will be coming over there. So are we clear, everyone? So we know that mitochondria has double membrane. It has its own DNA, right? And of course, it has its own ribosomes. That means that it can make their own protein. Then comes, it is a side, comes the function that Ritu is asking that, what is the function of mitochondria? It performs a most important function, that is the cellular respiration. It helps the cells to generate the energy and it produces the ATP. Are we clear, everyone, with this? Yes, mitochondria is called as the powerhouse of the cell. Why we call it as the powerhouse of the cell? Because it makes the ATPs. Yes, ATP, adenosine triphosphate. Many of you are asking me to explain the cellular respiration. So what happens inside our cells? Lot of chemical reaction occurs, right? Lot of chemical reaction occurs, and because of that we will see the production of ATP. right? We take oxygen, right? So oxygen will be going to the cell. And as it enters into the cells, a lot of reaction will occur. And when these reactions are happening, eventually what we will get? We will get the ATP. Are we clear? Everyone, are we clear? Cellular respiration, we will be utilizing the oxygen that you and I breathe. Very good. Divyashmi, ATP is adenosine triphosphate, is the energy currency of the cell. Very good. Are we clear, everyone? So we now know that mitochondria is a very important organelle, right? Repeating for those who are asking me to repeat again. We know that mitochondria is a very important cell organelle. It's a double membrane cell organelle, have its own DNA, have its own ribosomes, performs the cellular respiration and produce ATP. Very good Nishu. ATP helps in the active transport. Awesome. Yes, Bhakti, there were some issues over here, but now let's focus in the class. Very good. And over here, we know that, right, over here we have our ATP, and of course, it actually helps in the production of ATP. So the glucose that you and I eat, right, we get from the food, right, the glucose. After the breakdown of the carbohydrate moving right into the cell, and we will be getting the ATP. Are we clear, everyone? Yes. Just check the setting of your phone. Yes. Very good. Awesome. So we are clear with this, right? Sunday have explained it. Mitochondria, double membrane organelle, right, helps in the formation of the ATP and perform the cellular respiration. Very good. Awesome. I can see a lot of thumbs up over here. Very good, everyone. Let's quickly take up this question. Yes, over here we have the part which is present in, okay, over here the part which is present in the plant cells but not in the animal cells among the following is, yes, quickly tell me. Very good. That is the cell wall. We know that cell wall is present only in the plant cells and not in the animal cells. Very good, everyone. Very good. So we are clear with this, right? Now I can see Om Shankar is asking that, ma'am, can you explain how the mitochondria is performing the reactions that you will be learning in the 10th class? So we'll not be going into that detail, but I just did a session in the class 10's respiration, so you can check it out if you want to learn more. Yes, I'm not ignoring anyone, Gauri. Look at the smileys you are making. Yes, everyone be happy and we will be super excited kids, right? We'll be learning together. I want all of you to be very positive and, happy kids learn really, very quickly. Yes, look at these smileys that are coming. Awesome. Very good, Ayushi. Very good, Ritu. Yes, Sadhana will be discussing that. Yes, Nishu. Yes, Gauri. Yes, Mansi. Very good, everyone. Aryan, yes, I might have missed your name. Very good, Anushka, Lakshmi, Chitra. I can see lots of the correct answer. Divyashmi. Shivani is there. We have Aanchal, Shrutika, Shivani, Manas, Shreyansh. Very good, very good. See, you don't have to write call my name, call my name. Yes, let's move ahead, Ayushi is saying. Very good, Ayushi, for keeping us on track. Let's talk about the ER, right? Let's talk about the ER. Now, ER, can you quickly tell me? This is the most important and the easiest cell organelle. Yes? Quickly tell me. I can see, I think few of you have a little bit of slow internet. I can see you're answering now. Very good. ER, right? So, can you quickly tell me the function? I was just talking about that. Very good, very good. So we know that ER plays a very important role and there are two types of ER. We have smooth endoplasmic reticulum and we have the rough endoplasmic reticulum. Right? The question was, what is the function of ER? Very good. So we have it. Let's quickly discuss about it. So over here, we have the ER, right? Now ER, of course, we can have two. So we're talking about the first, which is the smooth endoplasmic reticulum. Over here, if you see, ribosomes are not present on it. It has a very smooth texture, right? Of course, if we talk about the structure of the ER, we have these network of membranes which are very close attached to the nucleus. And of course, we have these channels where the molecules will be moving. On smooth ER, we don't have any ribosomes and they help in the synthesis of the lipids. What they help us? They help in the synthesis of lipids and help in the membrane biogenesis. Yes, everyone, are we clear? Membrane biogenesis, of course, it's the process how the plasma membrane will be formed. Very good. Very good, everyone. I can see many of you are answering the right answer over here. Very good, Lakshmi. Very good, Shivani. Yes, Aditi. Yes, Bhakti, we will be discussing that. Mona Lisa, I think you have missed the class, right? Lipids, of course, plays a very important role in the making of our plasma membrane or the cell membrane. Bhakti, the name is endoplasmic reticulum. So over here we have the network, right? We have all of these, right? That's the ER, endoplasmic. We have it in the cytoplasm, right? Over here we have the endoplasmic reticulum. Membranes, network of membranes over here we have. Now many of you are asking about the membrane biogenesis. I've just explained. Can you quickly write? Very good. Yes, so let me just quickly repeat this once again for all of us. We are talking about the ER. It is of two different types. First, we are talking about the smooth ER, which of course plays a very important role in the synthesis of lipids, right? And plays a very important role in the membrane biogenesis. Now, membrane biogenesis is nothing but a whole process that actually helps in the formation of plasma membrane or the cell membrane, right? If you go back, if you recall, we know that cell membrane is made up of phospholipids. It has proteins and it has lipids. So lipids are coming from smooth endoplasmic reticulum and proteins will be coming from the rough endoplasmic reticulum. Ritu, why ER is smooth? There are two types, right? Smooth, ER is smooth because it does not have ribosomes. And then we have rough endoplasmic reticulum. It is rough because it has ribosomes on it. Can you see these blue color balls? These are the ribosomes and they are present on the rough endoplasmic reticulum. And because of that only they have a very textured, basically they are not smooth, right? Very good everyone. Yes, Vimlesh, we have cell wall only in plants and not in animal cell. We know that it helps in, of course, in the transportation, plays a very important role in the proteins and ribosomes are present over here. Right? Now, I want you to look here on the screen, right? Smooth like butter, yeah. Smooth endoplasmic reticulum. So we are talking about ER, everyone. Let's quickly recall. 2 points, important thing. We have two different types of ER. Smooth endoplasmic reticulum and rough endoplasmic reticulum. Right? Smooth ribosomes are not present. Right? And they help in the synthesis of lipids and membrane biogenesis. Smooth endoplasmic reticulum help in the synthesis of proteins and ribosomes are present on it. There's a second point that you're asking. They are the channel for the transportation. So as I have mentioned, they are very closely attached to the nucleus. So they help in the movement of the material from the nucleus outside into the cell. So whatever is coming from the nucleus, right, has to cross the ER. Mansi, I hope that you got this, right? Right, everyone? Everyone, quickly give me a thumbs up. I really want you to understand this. It is super important. Can't ribosomes stay independently? Om Shankar, they stay. There are ribosomes present, right? In prokaryotes also they are freely. Very good. In us also we can see that. But it plays a very important role. Very good, very good. Now let's quickly move ahead to the ribosomes. As you're talking about it, we have the ribosomes. Now ribosomes are the site, of course, we know already from the previous session of the nucleolus. We know that ribosomes plays a very important role in protein synthesis, right? They are present on the ER and they plays a very important role in the protein synthesis, right? Apart from that, they are the one that will be carrying the genetic information from the nucleus. Very good. Rafiq, you have to tell me who discovered RER and SER. Zarina, you also. Super easy question, right? It's a kind of a homework for you guys to look, go back and see that. Very good. Are we clear with this? Om Shankar, it's really very important because the message is coming from the endoplasmic reticulum only, endoplasmic reticulum, right? Very good. Awesome. So we are clear with this, right? These are the three important things. Let's quickly take up a question, right? The question is, which of the following cell organelle is called as a powerhouse of the cell? Kanika, you have, okay, I'll go back there just quickly. Yes. I can see the answers. Very good. Till the time you are answering, let me answer Kanika's question. She's asking about ER. ER are of two types. SER, on them we have no ribosome. RER have ribosomes. Proteins are produced. Okay, I'm a lefty, I'm just lying down over here, don't mind. And we have lipids production. Right, this is what we have to remember. And ER helps in the transportation, act as a channel of the transportation from the nucleus to the different parts, right, through different organelles. So basically it carries because it's very close to the nucleus. Awesome. Kanika, I hope that you got the answer, got the concept clear. And of course, the correct answer is the mitochondria. Very good, everyone. Now, can you quickly tell me which organelle? Very good, Ritu. Yes. Ribosomes, as I have already mentioned, Sandhya, they are the small units that are present on the ER, right? Basically, they are made up of the RNA and proteins. They plays a very important role in protein synthesis. Yes, please write your doubt. Ma'am, what happened to synthesize protein? What happened to synthesize protein? If proteins are not produced, our cells will not be able to perform, right? We call it as, yes, Rafiq, we do. Very good, very good. Now I was asking about that which of the organelle is called as the postman of the cell. Yes, Tanisha. RNA is the ribonucleic acid. Very good, Lakshmi, very good. Yes, Shreyansh, have answered your question. Golgi bodies, yes, very good. Sadhana, have answered your question. Postman of the cell, postman of the cell. Tanisha, very good. Smallest cell organelle, very good. I can see the answers. We are talking about the Golgi bodies, right? And as you know, the answer for this, can you quickly tell me who discovered the Golgi bodies? It's really very easy answer. And in which year? Very good, Chitra. Very good, Harsh. Very good, Neshu. Quickly tell me. I can give you a hint. It starts with C. Right? Very good. I can see the answers. It's I over here, so don't get confused. Yes. Very good. In the year 1898. Approximately during that time. Very good. So now we have the discoverer who discovered the Golgi apparatus. Now let's quickly discuss about it. Now can you look over here, everyone? Right, look over here. the structure of the Golgi apparatus, right, or the Golgi bodies. So if you look again at the structure, they are again in the network, they kind of stack onto each other, right? If you see this, see, this is not properly connected, but of course there are a lot of folds that we have in the Golgi bodies. So of course they are flat membrane bound vesicles, and they have cisterns stacked parallelly. So cisterns are nothing but these stacks that you see, right? We call them as the cisterns, and they are stacked onto each other. Parallel, very good. Very good. So that's a very important, that's a very important, right? So we have it. Very good, yes. Yes, absolutely correct. Vesicles are the small balls, right? These structures. These are the vesicles. Very good, Mayank. Oh, very good, Om Shankar. You're asking a very interesting question. Very good, everyone. So we are clear with it, right? Now we know the structure of it. Yes? Now let's quickly take a look at the functions. We know that they are the one that actually help in packaging. So you know what? ER will be sending the proteins and the lipids, right? It will be coming to the Golgi apparatus, right? And in the Golgi apparatus, packaging will happen, storage will happen, modification will happen, and the transportation will happen, right? So packaging is really very important, and transportation plays a very important role. Are we clear, everyone? And it plays a very important role in the formation of lysosomes. Yes, Shivani, you're absolutely correct. Post office of the cell is Golgi apparatus. Yes. Lakshmi, packaging of proteins and lipids. Very good, Monica. They help in forming the complex sugar from simple sugar. Lakshmi, I've answered your question. Proteins, lipids, etc. Very good, Rafik. Yes. Aditi. Very good. Yes, Venkata. Yes, Mayank. Thank you, Chitra. Supriya. Yes, Gargi. Tanisha. Very good. Yes, modification and packaging. Awesome. So we are clear with this, right? Yes, Anushka, I can see that you're answering. Very good. Mansi, you're taking notes. Very good. Aditi, awesome. I can see Lakshmi, Shivani saying clear. Awesome. So let's quickly revise. We're talking about the Golgi bodies, right? Now, Golgi bodies plays a very important role. They are the one that actually called as the postman of the cell. They help in packaging, transportation, and modifying the proteins, and will be sending to the different parts of the cell. What they are packaging or what they are transporting? Lipids and the proteins which are coming from the ER. Are we clear? They help in the synthesis or the formation of lysosomes also. Very good, everyone. Clear, right? I can see a lot of clear. And of course, now we are on the lysosomes. Sandhya, they are formed with the help of Golgi apparatus, right? Now let's talk about the lysosomes. Now, lysosomes plays a very important role, right? Right? So, of course, lysosomes are the membrane-bound sac over here, as you can see. Just single membrane-bound sac, inside which we have the hydrolytic enzymes or the digestive enzymes, right? And they are also called as the societal bags, right? Now, they plays a very important role. If something is wrong in the cell, for example, cell is not working properly, there is some infection or the cell is not functioning properly, lysosomes will burst themselves, right? They'll be releasing all the enzymes, and what will happen to the cell? Cell will die. So lysosomes are called as the societal bags of the cell. They help us in clearing out the worn out material in the cell. If an organ is not working properly, lysosomes can eat it up, can digest it. If a cell is not working, they'll release all the enzyme, and of course, the cell will die. Are we clear, everyone? Yes? Very good, Mayank. Very good, yes. That's why they are called as the societal bags. Mansi, have just answered your question. Sandhya, enzymes are the chemicals, right? Basically, we can call them as a catalyst that helps in the reactions that are happening. They will increase the rate of reaction, right? Very good. So for example, if you are learning, right, books are the enzyme that actually help you to learn further. In the reactions, we have the enzymes. Very good. So are we clear with this, lysosomes? So over here, can you see the cell, right? And over here, if you take a closer look, here we have, see? So whatever is waste that we have, right, will be getting broken down and of course will be released from the cell. Everyone, are we clear? Lysosomes have the enzymes, right? They have the hydrolytic enzymes. They have a membrane outside and of course inside they have the enzymes. Yes, yes, Sadik, they will die. What happened, Shivani? You're saying no, ma'am. Awesome. Very good. Yes, yes, yes. I can see clear answers. Very good. Sandhya, autolysis means killing themselves. Automatically the cell will just burst, and the cell will die. Please write your doubt. I've answered your question, Sandhya. Stop spamming. Yes. Awesome. Let's move to the next important question that I would be asking you. And over here we have the question, which of the following cell organelle is called as a suicidal bag? Super easy question. Pearl clear, Kanika. That's amazing. Very good. B, I can see. Very good, everyone. Yes, and the correct answer is C only. We know that lysosomes are called, right? We know that lysosomes are called as the, what? As the suicidal bags of the cell. Very good. So we know that lysosomes are the suicidal bags because they are the one, if they have the lot of hydrolytic enzyme, if the cell is not functioning properly, they will release the enzyme and the cell will die. Very good. So are we clear, everyone? Yes, Sandhya, this topic is not related, but you just keep on writing. So let me just tell you. So that's a way the different cells we have, specifically if you take an example of the WBCs, that's how they'll be engulfing or killing the cell. Sandhya, to your question. So, Sharanya, a lot of waste material is generated in the cell itself, right? So a lot of reaction occurs, a lot of waste material comes. So it just comes out, right? And that can be taken care with the help of the lysosomes. And apart from that, a lot of organelles that will be worn out because of that. Very good. So let's quickly move ahead, everyone. Let's talk about the vacuole, right? We know that vacuole plays a very important role there again. Membrane bound organelle. I hope that you are paying attention to these words because these are important. They are the membrane bound organelle, right? They plays a very important role in the storage. They are the one that store, and because they occupy a larger space in plant cells, they provide the turgidity and the rigidity. Now vacuoles are also present in the animal cells, but they are really very small. And in plant cells we have a large vacuole, right? So that's a major difference between the vacuole which is present in the plant cells and in the animal cells. Shivani, you're asking how lysosomes are made up of. How you are saying how they are made? So they are made with the help of the Golgi bodies, right? So what happens? We know that lysosomes are the enzymes. It has the enzymes, right? So of course, when the information that comes from the ER goes to the Golgi bodies, and of course there are vesicles formed, and that's how they are formed. Very good, right? Stored biomolecules. Now, all the biomolecules that we have, right, could be anything. For example, it is storing all the ions that we have, all the important materials, all the elements, right? Whatever is important for the functioning of the cell is stored in the vacuole. Now, Sadik is asking a very interesting question. Vacuole is a double membrane. What is written over here? It's a membrane-bound structure. That means that it will just have a single membrane, right? Very good. If you talk about the vacuole more further, we know that plant cells have the large vacuole. Very good. Are we clear with this, everyone? Got it? Very good. Yes? So everyone, let's just quickly recap what are the things we have studied. We have talked about the mitochondria, which is the powerhouse of the cell. We have talked about the ER, which plays a very important role in the synthesis of lipids and synthesis of protein. We talked about the Golgi bodies, which actually plays a very important role in the packaging, storage and modification of the proteins and lipids, and of course send it to the different parts of the cell. We talked about the lysosomes, which contains the hydrolytic enzyme or the digestive enzyme that actually help in clearing out of the cell. We also call them as the suicidal bags because they are capable of killing the cell. And now we are discussing about the vacuole. Vacuole plays a very important role in the storage. They are the storage sac, and they are the one that store different types of biomolecules that are required and that are really very important for the various reactions that occurs in the cell. They are bigger in plant cells and smaller in the animal cells. Are we clear, everyone? Yes? Are we clear? Turgidity means a stiffness, right? Yes, Aditi, we have discussed in the cell wall. Very good, Gargi. Very good. Now let's quickly talk about the plastid because I can see plastid is in demand. So we know that there are different types of plastids. We have leucoplast, chromoplast and the chloroplast. Now let's quickly discuss about the leucoplast. Now leucoplasts are present in the non-photosynthetic part like roots, right? They are white and transparent plastids and they are present in the roots like where of course no photosynthesis occurs. And of course they actually help in the storage of the starch or the oil granules, right? Very good. Colorless cells, absolutely correct. No pigment is present. Very good. So this is important information that we have to remember about the plastids, right? Especially the leucoplasts. They are white and transparent plastid and what they store? They store the starch or the oil granules. Then of course we have the plastid and we'll be discussing about the chromoplast. Now chromoplasts are colorful plastids. Very good. Very good. Now chromoplasts are the colorful plastids, right? And they give the colors to flowers and beautiful flowers and fruits that we see. Now I'm sure you must have seen tomatoes. We have beautiful flowers, right? All of these get the color from the chromoplast. That's a very important pigment. Very good, Tanisha. Yes. Yes. I think you're mentioning ribosomes. They help in the protein synthesis. Very good. Now comes the last plastid, which is the chloroplast. And of course, this gives the green color, right? And of course, it helps in the performance of the photosynthesis. It's because of chloroplast only, right, which have the chlorophyll. We know that. We have the green color, right? And of course, it has a green color plastids and plays a very important role in the photosynthesis. We know that photosynthesis is a very important process by which plants will make their own food. And over here, we have a very quick summary of the photosynthesis. Photosynthesis, plants will be taking the carbon dioxide from the atmosphere, water from the roots, and in the presence of sunlight and in chlorophyll, they will be making glucose and releasing the oxygen. Yes, Mithun, we will be discussing about cell division, but not today. Yes, we'll be discussing in some another class. Chromoplast, Mansi, will be giving the colors, right? Colors to the fruits and flower. You're asking what is the important function of the leucoplast? So leucoplast helps in the storage of the starch and the oil granules. So oil, the fat that we have, right, in the plants, that will be stored in there. Yes, Vartika, I've answered your question. Awesome. Let's move ahead, everyone, to a very quick question over here. Which plastid stores oil granules, right? Very easy question. Right, quickly tell the answer. Which will be storing the oil granules, right? Very good. Please quickly answer. I can see the answers already. Very good. Quickly answer, everyone. I can see the answers and all of you are going for A. So the correct answer over here is, of course, the option number A. That is the leucoplast, right? Leucoplast. Awesome. Very good, everyone. Are we clear with this? Awesome. So with this, we have over here, we came to the conclusion right over here. And that's a summary for all of us. Now I can see there are still few doubts you have. Thylakoids, Mansi, yes, chlorophyll contain the thylakoids. Please write your doubt. I can see someone is asking, I think Chitra is asking about the tonoplast. Right, a tonoplast, of course, it's a membrane. Basically, we can say that when we talk about, see over here, it is related to the vacuole, right, chief vacuole. Very good. Achal, I've answered that question. Yes. Tanisha, we're talking about the endocytosis. Now, endo means the things will be moving inside, exo means the things will be moving outside the cell through the plasma membrane. Thank you, Ritu. Granules are these small structures which contain the oils or the fats. Enzymes, Prem Kumar, enzymes are basically the chemical catalyst. We call them as a catalyst. They actually help in increasing the rate of reaction. You will be learning more about them in your upcoming classes, right? I'll not be able to explain you more because it is difficult for us to contain that information in this class. Yes, are we clear, everyone? Very good. Please write a doubt if you have. Biomolecules, Rafiq have mentioned. Lipase, Gargi is an enzyme that helps in the breakdown of lipase. That we'll be learning in 10th class. Very good, everyone. I think, are we clear? Right, so we talked about the tonoplast, which of course is a membrane that will be covering the chief vacuole over here. We learned about the plant cell and the animal cell and the different organelles. And I can see a lot of you are asking about the homework question. So over here we have the homework question. There are two important homework questions. Now you have to do a bit research over here. We have to write the difference between a plant cell and the animal cell, which is a common question asked in the examination. Then you have to find out that which organelles have double membrane bound, right? Which of these have double membrane, right? And then write their function. And you can find out which of the organelles have the single membrane bound structures also. Yes. Right? It will be great if you write the answers there. Right, everyone? So we have discussed a lot of things. I know that there are so many of you and there are things that, maybe I've missed some of your questions. So I would request you to write the questions in the video, right, in the comment section below. Right? We will be taking up those or we will answer there. So if at all, if you have missed any question over there, please share it with us there. And of course, in next class, we will be meeting on 2nd of June and we will be talking about how to make the plant and the animal cells. Super interesting question, right? And of course, we have got you covered. Don't worry. And hit the like button, everyone. That really means a lot. It means a lot. Share with your friends and subscribe. I hope that you have subscribed to our channel. Yes. Hit the like button, everyone. That means a lot. And of course, there's a free class trial link. You can definitely try that. Yes, Tanisha, it is. Sandhya, you have asked so many questions. I feel that this class will not be able to, cater. You can watch a video which we did on the cell wall. It will be able to help you. And it's a very easy question. Do some research, you will be getting the answer for it. Very good. Om Shankar, it's a very complicated questions you have asked. Think about it. So it involves the ER, it involves the Golgi apparatus. So there's a hint and you can read more about that. We have all the information. I'm sure you'll be able to get the answer for that. Thank you so much, everyone. I hope that you've enjoyed the session, right? And I hope that you have taken the notes of the all important points that we have discussed. We will be meeting on 2nd of June and get ready, everyone, right? And see you. Lots of love to you. Bye-bye, everyone.